Today I am going to discuss about the heme synthesis process. Heme is an iron containing compound. The amount of iron contained in a human body varies between age and gender groups. There is a distinct difference between male and female in the amount of iron present in the body. This is due to the less number of RBCs, amount of muscles, and liver and small size of the liver in females compared to men. Men have around 50 milligram per kilogram of iron, whereas female has only 40 milligram per kilogram iron. Almost all iron in body is incorporated into heme containing proteins, particularly hemoglobin, myoglobin, and cytochromes. Literally, there is no free iron in your body except while it is absorbed from the intestine. The unique properties of heme allows it to function both as an electron carrier and as a catalyst for redox reactions. An iron molecule is coordinated within a tetrapyrrole ring, that is the structure of a heme compound. Biochemical pathways of heme synthesis involves many steps and many enzymes, and it happens in two locations. A part of the reaction happens inside the mitochondria. Actually, it starts in the mitochondria and ends in mitochondria. In between, some of the steps happens in the cytoplasm. And a deficiency in any enzyme, different enzymes are involved in synthesis of heme. In a deficiency of these enzymes or substrates will lead to the accumulation of certain intermediates, which will lead to the formation of certain clinically significant disorders. So it's a lead to a group of disorders called porphyrias. These are hepatic or erythropoietic. And because heme synthesis happens in the bone marrow as part of the erythropoiesis, formation of RBCs, and also in the liver as part of the synthesis of many metabolic materials, including certain enzymes containing heme. So, these porphyrias are hepatic or erythropoietic, depending upon where this accumulation is happening. And also, these porphyrias are acute or chronic, and they cause many neurological dysfunction, mental disturbances, or photosensitivity. And also, the different symptoms include change in the urine color, then abdominal pain, abdominal colic, highly agitated state, tachycardia, abnormally high heart rate, and respiratory problems, and also nausea. Heme synthesis happens through a highly conserved pathway, which involves both cytosolic and mitochondrial compartments. As I said before, the starting point of heme synthesis occurs in the mitochondria. Then the intermediate product moves to cytoplasm, certain changes happens, and the later stage, the product will move back to the mitochondria, and the last stage of heme synthesis happens on the inner mitochondrial membrane. And during this process, protoporphyrin 9 is generated, which is an important compound in heme synthesis. And all the heme biosynthetic genes are nuclear encoded. So they are present in the nucleus, and the mRNA is produced in the nucleus, and which will be moving to cytoplasm, and translation happens in the cytoplasm. Then the location, as I said before, most of the heme synthesis takes place in developing red cells in the bone marrow. And about 15% of the daily production of heme takes place in the liver as part of the formation of heme containing enzymes. There are different heme containing enzymes like catalases, peroxidases, cytochrome, P450s, etc. In the liver, heme biosynthetic enzymes are turned over rapidly depending upon the metabolic necessity. Sometimes you may need some of these enzymes in a higher quantity. So the liver synthesis of heme in the liver will increase. So drastic increase can happen, or similarly, if there is a decreased necessity, the synthesis can decrease. Whereas in erythropoietic tissue, that is in the bone marrow, which maintain a steady state of heme synthesis. There is no sudden increase or decrease in the synthesis of heme in bone marrow. And the regulation of heme, whereas in liver as well as in erythropoietic cells, is depending upon the availability of iron. So it is very important to have enough amount of iron in your food. Porphyrins are large heterocyclic organic ring structures. This composed of 4 modified pyrrole subunits, 1 subunit, 2, 3 and 4 subunits, and they are connected by methane bridges, that is double bond C single bond. So like it's double bond C single bond. So they are connected. These 4 pyrrole molecules are subunits are connected by methane ring bridges. It's an example of naturally occurring porphyrin. Heme in biological system consists of Fe2 plus. Fe2 plus at the center of this heme molecule, complexed with the 4 nitrogen of the porphyrin molecules. 3 structurally distinct hemes are present in human body, heme A, B and C. It's critical for biological functions. So several enzymes, example cytochromes of oxidative phosphorylation, cytochrome P450 family. So the first step in heme synthesis happens in mitochondria. And this is a condensation reaction between succinyl-CoA and glycine by pyridoxal phosphate and catalyzed by the enzyme delta aminolevulinic acid synthase. As a result, the delta aminolevulinic acid is formed. And this is a rate limiting reaction in heme synthesis. So the rate of production of delta aminolevulinic acid determines the rate of synthesis of heme. So if your body needs to synthesize more heme, more of this reaction happens. And if the body's requirement of heme is lesser, this reaction speed will be reduced. So this is a rate limiting step in heme synthesis. So this enzyme, this aminolevulinic acid synthase, ALS, there are 2 types of ALS, ALS1 and ALS2. ALS1 is ubiquitously present. It is present always. Whereas ALS2 is not present always. It is in erythropoietic tissues alone. And its rate will be determined by the need of erythropoiesis. Then, once this delta aminolevulinic acid is formed, this will move to the cytosol, where 2 ALA molecules are dimerizes, forming porphobilinogen in the presence of the enzyme aminolevulinic acid dehydratase or porphobilinogen synthetase. So ALA is transported to cytosol, dehydrated in presence of the enzyme ALA dehydratase. There is a dimerization of 2 molecules of ALA and formed porphobilinogen. Then this porphobilinogen, 4 molecules of porphobilinogen condense in a head to tail fashion, forming a tetrapyrrole. So containing 4 pyrrole molecules. That's the name of the molecule is hydroxymethyl bilane in presence of the enzyme porphobilinogen deaminase. So this hydroxymethyl bilane has 2 fates. One is decided by the enzymatic action, and other is non-enzymatic action. So this is the tetrapyrrole molecule, that is hydroxymethyl bilane. 1, 2, 3, 4 molecules. So this hydroxymethyl bilane is enzymatically converted into uroporphyrinogen 3, and the enzyme is uroporphyrinogen synthase. Okay, there is a negative feedback here. When there is more uroporphyrinogen 3, what will happen? The synthesis of or the conversion of this hydroxymethyl bilane to uroporphyrinogen 3 will be suppressed. So the amount of this uroporphyrinogen in the body decides the speed of this reaction, speed of its formation. So that is a negative feedback. More uroporphyrinogen present, the speed of reaction decreases. this uroporphyrinogen 3 is decarboxylated by uroporphyrinogen 3 decarboxylase. So the next is a decarboxylation reaction, and the formed coproporphyrinogen 3. So uroporphyrinogen 3 decarboxylation happens and forms coproporphyrinogen 3, and this will move to mitochondria. So this compound, this coproporphyrinogen 3, is most important in heme synthesis, which moves to mitochondria and further reaction takes place there. So next step is the formation of the conjugated ring. So the propionate residues of coproporphyrinogen 3 are decarboxylated and forms protoporphyrinogen 9. So this compound is formed and by the decarboxylation. Later, then what happens? An oxidation step happens, catalyzed by coproporphyrinogen 3 oxidase, forming protoporphyrin 9. So this is formed, or proto 9 is formed by this oxidase enzyme. This oxidation requires oxygen as a terminal electron acceptor. So oxygen is used as electrons are released and oxygen is the electron acceptor. So here there is a removal of or loss of 6 protons and 6 electrons and produce a complete conjugated ring is formed. So once this ring is formed, then comes the final step in the heme formation, that is the insertion of iron molecule, Fe2 plus molecule, and ion into the molecule. So final reaction heme synthesis takes place on the inner surface of the inner mitochondrial membrane. So that is the location of the final reaction of the heme synthesis, inside of the inner mitochondrial membrane. So insertion of Fe2 plus into the ring system happens by the help of the enzyme ferrochelatase, and the heme molecule, the complete heme molecule is formed, and the insertion of this ion causes or produce red color to the heme molecules. So this is how heme is synthesized in the body. Thank you. So you may answer 2 questions. And what is the rate limiting step in the heme synthesis? And also there is a negative feedback control mechanism existing in the pathway, in the heme synthetic pathway. And what is that, or where is that negative feedback mechanism?